Thank you very much. I'm going back to an very old story at the beginning, that of DNA. DNA is, of course, the origin of memory, genetic memory, since a very long time, perhaps from the beginning of life, of organized life, 3.5 billions of years. But I like to show you that maybe this is not the end of the story, and DNA exists also because of water. And I'd like to show this morning some recent work we have done showing that water is not only a solvent, it's also a polymers which could interact with the DNA, helps the DNA, and also the information from DNA to water can exist through the origin, the emission of very low electromagnetic waves. So, just to remind you that the story of DNA discovery is very long, starting by the first evidence that bacteria characters could be originating from DNA, Avery, MacLeod, McCarthy. And then, of course, came the very important finding of the double helix structure. But I like also to emphasize that since that there are many important steps, and most of the steps have been achieved by Nobel laureates, some of them are here. But I like to stress the importance of DNA sequencing, the reverse transcription by retroviruses, the restriction enzyme allowing the cloning, of course, and also the polymerase, more recently, the polymerase chain reaction by Gary Mullis, who got the prize, the Nobel Prize in Chemistry in 1993, and followed very soon by the use of a polymerase, a DNA polymerase highly heat resistant from thermophilic bacteria, the Taq polymerase. And then came, of course, the sequencing the human genome and the high technology to sequence this high-speed DNA. How I came to this field and what I like to add to something to those past discovery came 10 years ago when I did a very strange experiment using a small bacterium called Mycoplasma pyrum. You can see here its shape. This mycoplasma like to live at the surface of human lymphocytes. Actually, it's a frequent companion of HIV, at least in the in vitro studies. This mycoplasma has a very small size, but of course larger than the virus itself. It's about 300 nanometers in size. And I decided very simply and very naively to filtrate, to separate this mycoplasma from the virus by using filters. And first, I made a controlled experiment using only the mycoplasma, infecting lymphocytes, no virus, and filtrate in a filter we should remove all the mycoplasma. And indeed, when we use the filtrate after 0.02 micron or 0.1 filter to grow the mycoplasma in acellular medium, SP4, which is a classical medium for growing mycoplasma, the filtrate was sterile, no growth at all. But if I put the same filtrate on a culture of human lymphocyte, of course controlled for not having any kind of mycoplasma infection previously, the surprise was after 2-3 weeks, we got the mycoplasma back. And I did this experiment, and my associate also, many times. It was not an artifact of the filtration, for instance. We also checked for by PCR, a nested PCR, which is a technique which can amplify millions of times one sequence of DNA. We use a gene we have previously cloned called adenosine, the adenosine gene, the gene which allows the mycoplasma to bind the lymphocytes. No signals. So this led me to think maybe the genetic information of mycoplasma was still in the filtrate, not under the form of DNA, but under the form of something else. And 5 years ago, I tried to investigate with some my associates the possibility that some forms of water could be involved in this. And we came across a very surprising thing, which is the fact that deletion of that DNA or the mycoplasma as a microorganism could emit some very low frequency electromagnetic waves in high dilution of water. So the equipment at the beginning is very simple. We put the sample on a sensor coil in a plastic tube in a solution, and we amplify the signals, the electric current coming from the magnetic part of the waves. The electric current is then analyzed on a computer with appropriate software. And the surprise that we could also detect similar signals with many filtrates of microorganisms, virus and bacteria, or also from the plasma of human people infected with the same agent. So I'm going to describe you the principle of each experiment receiving the signals. We have to filtrate. It's not just to remove the initial microorganism, probably to dissociate some structure in the water. We have to filtrate. Then we have to dilute out, and only in some dilution, you see in yellow, the high dilution are producing the signals. And we use, first we use electromagnetic background of very low frequency, 7 Hertz to 100 Hertz, and we get signal of slightly higher frequency, 1,000 to 3,000. And of course, we have several ways of recording. You can see the waves over the background, which are easy to detect. This is a crude representation of the amplitude of the signal, which is also higher over the noise. And we can do some Fourier transformation to analyze the peaks produced. Here is background noise, in which in yellow you see the 50 Hertz coming from the electric power, which is always there. We have to realize that we are always exposed to this very low electromagnetic field. And when we got a positive signal, we see a shift to higher frequency. Of course, there are several means to quantitate this, and also I show you here on the left the background, on the right the positive signals, and on the bottom you see that we get several harmonic peaks. But I have to tell you first that this is not specific of a specific DNA. We get this kind of signals depending on the background. They are all similar for all bacterial DNA or viral DNA. But we cannot exclude that using more refined technology to capture and analyze the signals to see some slight differences according to the DNA species. Of course, we investigate the nature of the structure which could produce those signals, and we found that they are resistant to all kinds of enzymatic agents. It's not DNA, it's not RNA, not sensitive to detergent, to acidic pH, to ion concentration, even to ethanol. But the structures are rather heat sensitive. They are destroyed by freezing and also by magnetic field. And using filters, we could discriminate between viral-induced nanostructure, which are smaller, less than 20 nanometers. Remember that the virus range, the virus size is between 150 and 25 nanometers. And for bacteria, we could find a size slightly higher between filtration or remaining after filtration by 20 nanometers, but suppressed by filtration at 100 nanometers. So those structures were found also rather stable in our hands, in our solution. And of course, we investigate the relationship with the microorganisms from which they were originating from. And it was also very clear that there is no linear correlation with the number of microorganisms present before the filtration. For instance, we use Escherichia coli suspension, and even with 10 to the 9 or to 10, we had the same similar signals. I must say that this problem fits with the idea that we are dealing with a non-linear phenomenon, perhaps with a quantic phenomenon. We found this emission from many pathogenic bacteria listed here. It's not, of course, the list is not complete. Also from some virus, we haven't completely investigated all the viral families, but of course, interested by what's happening in HIV and AIDS, also influenza, hepatitis B, and hepatitis C virus. And in some example, we found that one gene only was sufficient, or even a fragment of gene of DNA was sufficient to induce the signal. This was the case of the adenosine gene we had cloned from Mycoplasma pyrum, and also I'll show you later for some HIV genes. So, of course, this implied that probably some conformational DNA is involved in this emission, but not excluding the possibility that this is a general phenomenon occurring for all kinds of DNA, but we are just picking up the tip of the iceberg, the sequence which are more emitting than others. Condition for EMS adduction is very strict in that we have to filtrate, we have to use high dilution in water, we have to use mechanical agitation between each dilution, and we have to excite the system by the ambient electromagnetic background, starting very low frequency, at 7 Hertz. And this is prevented by some alloy like mu metal, which will absorb the waves. And this is a resonance phenomenon. The energy comes from outside, from the electromagnetic background. And of course, now we are using a very specified excitation waves, starting with 7 Hertz. This has been described in more detail in a paper we have published last year. Now I come to the theoretical explanation of this. Of course, this is just preliminary. We have to talk with physicists, and we have simply to give them the facts so they can work on those observations. But it seemed to be clear that DNA is very important first, and we know from the very first work on X-ray diffraction of DNA by Wilkins and Franklin that there are water molecules strongly associated with DNA, and they are contributing to the stability of the molecules, and also they can interact with the bases to make new hydrogen bonds. There could be hydrogen bonds in water. So our idea is that perhaps this atmosphere of molecules, of water molecules, can enlarge and form large aggregates in the water solution. And indeed, the physicists are telling us that water molecules can form large polymers by hydrogen bonds. Now, they are very short-lived. However, more recently, stable structure can emerge as quantum coherent domains. This is the theory of Emilio De Giudice, which could be self-maintained by the EMS emission upon excitation by natural waves. So where those natural waves come from? Geomagnetics, but also the Schumann resonance. Schumann resonance were discovered by Otto Schumann in this country, in München, when you measure the waves which are guided between the surface of the Earth and the plasma of the ionosphere. And those waves are produced by lightning. So at any time, any point of the Earth, there are always lightning. And this lightning maintains a circulation of waves around the surface of the Earth. And those waves start at 7 Hertz with a multiple of this, but they are very low frequency. We can detect them in our system. Also, those can change with also the solar wind, but the most important thing for us now are human activities, because we are living in a world of waves. We use waves all the time, but high frequency waves. Here we are dealing with very low frequency waves, which have very low wavelengths, 1,000 kilometers. Another possibility, another theory, that since we are shaking very strong the dilution, we are forming nanobubbles with the gas, and this could also modify the structure of water at the interface. So our current interpretation is the following. So we get in a low dilution formation of structural crystal-like gels like this. They are not moving, but if we dilute them out, we see this type of structure, perhaps induced also by the electromagnetic waves of the background. And if we look at the possible orientation of water molecules, it's quite possible we have a laser-like oriented system of activation of water molecules in electromagnetic field. And you can see on the right the explanation of the Giudice, in which we are forming in water structures which are, of course, surrounded by non-coherent molecules, but those coherent structures are self-maintained, perhaps by the electromagnetic emission. So I went further. If I come back to my first experiment in Mycoplasma pyrum, we should imply that the structure induced in water maintain in some way their genetic information of DNA. So I'm going to show you very recent experiments, but we have done them several times, 8 times at least, and they are all yield the same phenomenon. Yes, for at least some short DNA sequence, we can reproduce this sequence in water. So I show you what we have done with HIV. This is one of we won't be surprised, one of my favorite objects. And HIV DNA has a very complex structure, as you see here, but you can see at both ends, this is what we call long terminal repeat, which contain the sequence relation for expression. So what we did, we used the sequence of the LTR to make short DNA amplification by PCR. And you can see here both a rather large fragment of DNA starting at the top and going back to almost the bottom, LTR out. And then inside this amplification, we secondarily amplify a shorter segments which correspond to the in primer, LTR in at the bottom. And this is a sequence 104 base pair long, containing mostly a region of the DNA called TAR. TAR means transcription activated region, which is bound by another proteins, by a protein of HIV called TAT. And this binding allow, with a complexation with a cellular kinase, allow the extension of all messenger RNAs. So we use this sequence for our experiments, which is described here. So we first diluted the TAR, so-called TAR DNA in water. We had, of course, some signals coming from dilution. So we choose one tube of dilution -6, for instance, or -4, and this was put in a container of new metal to insulate this from the external electromagnetic background. And of course, we inject a frequency of 7 Hertz in a coil. So this induces a magnetic field, and inside we put our dilution of the tube close to, but not completely attached, to another tube of water. And actually, we could have a distance of 10 centimeters between the two tubes, so the one that touched the other. We then generate the current, and we had to wait 18 hours. If we are just 1-2 hours, it's not sufficient. This is a very low process. Then we take the tube of water, we dilute it out, and found again the EMS, the signal production, in some dilution, of course, which are lower because we start from -6. So now -2 from -6 means -8. So we had positive tubes, and of course the question was this information transmitted by electromagnetic waves from one tube to another containing the DNA information. So we did some very specific PCR using the primer as described before, and analyzed the DNA produced in electrophoresis. This shows the bands. And on the top, you see the donor tube, and there are dilution, -2, -3, -4, not surprising. But on the bottom, you see the DNA bands formed in water, and they go far up to 10⁻⁸. And we check by sequencing, by cloning the DNA and sequencing it. It was really LDLTR HIV DNA. Same sequence, up to 98%. There is only 2 nucleotide difference. So I think we have shown in this experiment, we have repeated many times, that at least for some sequence, short sequence of DNA, we can transfer the information into water. So this opens, of course, a large field of exploration. I like to do with physicists, of course, to explain this, but it's clear that water is not simply a solvent. It's maybe much more in our life. Now, the last part I will go quickly is on medical application. We can detect those signals coming from the plasma, the fresh plasma of patients infected with some infectious agent, which is not surprising. But for Borrelia, for instance, it's important because Lyme disease caused by Borrelia burgdorferi is a chronic disease, and we have some difficulty to detect the presence of Borrelia in chronic infection. We can do it easily by our technology. But more interesting is the case of diseases which are not known to be of infectious origin, like several neurodegenerative diseases, Alzheimer, Parkinson, multiple sclerosis, various neuropathy, chronic Lyme syndrome, autism, at least some cases, rheumatoid arthritis, even some cancers. So, of course, the idea is that perhaps those signals are coming from traces of infectious chronic infection by bacteria or viruses. So, of course, here the objective is clear, is to identify the bacterium or the virus involved, which might come from our own gut, actually. But I'd like to terminate by the special case of HIV. Indeed, in patients infected with HIV, we could not detect very much the signals when the virus load in their plasma was high. On the contrary, we could detect very regularly signals coming from HIV DNA sequences. They are perhaps integrated. They may come from fragments of proviral DNA released by dying cells, possibly in the circulation, but they could also represent some kind of episomal DNA replication. You remember certainly that the treatment of HIV has made many advances, We can use nucleose, the reverse transcriptase inhibitors and also protease inhibitor, and their combination can keep most of the patients alive. They don't cure. The patients are still a reservoir of virus. If we treat, we reduce that reservoir, but if we interrupt the treatment, the reservoir induce again a highly active application, and we lose the advantage. So what we found regularly in the patients which have no virus load in their blood, signals coming from DNA, and that DNA we analyze more classically by PCR, is made of several genes like POL, the polymerase gene, but also the long-term NLRP, the LTR. So whatever the explanation, we have now a biomarker for detecting the virus which remains after the antiretroviral therapy. So this opens the way to target this reservoir by complementary treatments, complementary to the antiretroviral therapy. This has been also recently published, and I'd like now to finish by saying that we are just exploring a new field, DNA water waves, which could lead to some medical application, increase the blood safety, preventing nosocomial disease, especially after heart surgery. We can detect signals coming after heart surgery from the fact we are inducing ischemia. Probably in the gut, our mucosa is suffering and release some microbial products into the circulation. We can also, as I said, look for chronic diseases. The role of microbial agents could be one of the factors of those diseases. I'm not saying they are all the cause. And for HIV, of course, use this as attempts to eradicate the infection. I'd like to thank my collaborators in those studies, and I should add also Claude Lavallée, who was involved in the latest experiment. And thank you very much.